Hi, I'm Dr. Anandi. Welcome to my channel Make Biology Simple. Today we'll discuss about the glycogenesis. It's one of the carbohydrate pathway. Glycogenesis is a polysaccharide synthesized in the cytoplasm and it's stored in liver 10%, muscle 2%. These are the steps. Glucose is converted to glucose 6 phosphate and 1 phosphate, and then it is produced with the UDP glucose. It's called activation of glucose. Then it is elongated with alpha 1,4 glucosyl units. Then the branching chain starts, that is alpha 1,4 to 1,6 glucosyl units. So these are the steps involved in the overall view of the glycogenesis. Genesis means synthesis. Glycogen means glycogen is synthesized. So this is the steps involved in the glycogenesis. Next we'll see about the branching. So actually the glucose will join to a primer. That primer is called a protein. It's called glycogenin. To the glycogen only the activated UDP glucose will transfer and donate the glucose to it, and it's attached to the glycogenin's primer, and this is how the elongations takes place, and then finally branching occurs. So this is the overall view. You can have an idea about how an addition of glucose is attached to the UDP, and then it is attached to the glycogen. Blue color is glycogenin and the rose is a new additional glucose is attached to the elongation of the glycogenin. Next we'll see how it has been branched. So once a length of say 10 to 16 glucose units are joined, and then a portion of it is cut and it is now elongated with the alpha 1,4,1,6 glycosidic linkage. So this is summary. Glucose is converted to glucose 6 phosphate with the help of glucokinase, and the phosphate glucomutase to converted glucose 6 to 1 phosphate, and glucose pyrophosphate will convert glucose 1 phosphate to UDP glucose. This is the extension of the UDP glucose to the primer called glycogenin. The enzyme is glycogen initiator synthase. Then glycogen synthase will elongate, will keep on adding the glucose to the primers. And after that a branching starts. The branching is very important. The branching enzyme is called glucosyl alpha 1,4,1,6 transferase. So this is how a branching starts, and the polymer glycogen is produced in the system, that is for example in the liver and the muscles. So that's all about the basic process of glycogenesis. Thanks for watching my channel Make Biology Simple.